Everyone, I'm so happy to be able to give you this update today. So I guess I'd like to tell you about what I've been doing in the last 6 months. And what you remember is that I'm working on the stem cell project to turn stem cells into insulin producing cells. And what I haven't told you before is that it's a 6-stage process. So from beginning to end, it takes 6 distinct steps. And what I worked on was stage 3. I was really trying to improve the derivation of stem cells into stage 3 type cells that would eventually become insulin producing cells. And I did that using a heart drug called AAP10. So AAP10 is super cool. What it does is it looks at these proteins that I use that are called connexin 43, which acts sort of as like a door between cells like this. And they can be closed or they can be open, and AAP10 holds the cells open like this so that everything can whoosh through in between these two cells. So what I found is that when I had the open proteins like that with the drug, I got a major increase in the number of stage 3 cells that I could derive in a culture compared to when there wasn't any peptide at all. And then the really super awesome thing that I found after that was even when I took this peptide off, so even when my protein was able to open and close as much as it wanted to by itself, I still got an increase in the number of cells that are likely to become insulin-producing cells. So I wrote my whole thesis actually on the application of this peptide on my protein, connexin 43, and how it's helping us improve our outcome of cells that we can eventually use for transplant. And this is really, really important because a lot of people can do these different differentiations. A lot of people can turn stem cells into insulin-producing cells, but we really think we found a way to do it better and more efficiently. And I hope you're all doing well out there, and thanks again for donating to the project.